Hello everyone, I am a researcher at the National Center for High Performance Computing. In this series, we will explore how raw 16S sequencing reads can be transformed step by step into interpretable results about microbial communities. The experimental details may vary from sample to sample, but the core logic of the analysis is much the same. First, we obtain 16S amplicon sequences. We then perform quality filtering and denoising to infer reliable sequence variants, known as ASVs. Only after that can we describe community composition and compare differences across samples. We often use QIIME2, usually pronounced QIIME2, to carry out this analysis. One of its most important features is traceability. It is like creating a complete production history for the data. In QIIME2, an artifact is a data object that can be used in subsequent analyses and usually has the file extension .qza. An action is a processing step performed on the data. A visualization is a file used to view results and usually has the extension .qzv. Provenance also records the input data, parameters, and analysis history, allowing us to go back and confirm exactly how each result was generated. Let us first clarify what questions 16S amplicon sequencing can answer. It uses polymerase chain reaction, or PCR, to amplify the microbial 16S ribosomal RNA gene rather than sequencing the entire genome. This is somewhat like identifying different groups by using a specific set of distinguishing features. It helps us describe which microbial groups are detected in a sample and how community composition differs among samples. However, the number of reads is not the same as the absolute number of cells, and 16S results cannot directly provide a complete picture of which functional genes the microorganisms possess. In experimental design, primers affect which microorganisms we can ultimately observe. The 16S gene consists of alternating conserved and hypervariable regions, with hypervariable regions usually labeled V1 through V9. For example, the commonly targeted V4 region can be amplified using the 515F and 806R primer pair. If the primer binding sites of a microbial group are a good match, that group will generally be amplified more easily. If there are mismatches, amplification efficiency may decrease. As a result, the community composition we observe is also influenced by the selected target region and primer bias. In addition to sequence data, we also need sample metadata. This table records background information for each sample, such as the experimental group, participant, time point, and batch. It must be linked correctly to the sequencing files through a unique sample ID. These fields are required for coloring plots, comparing groups, and building statistical models later in the analysis. If sample IDs are matched incorrectly, or if essential fields are not recorded when the experiment begins, no matter how sophisticated the downstream algorithms are, they cannot recover the missing study design information. Newly obtained raw reads cannot be treated as reliable biological results because they may face three types of problems at the same time. First, quality may gradually decline toward the ends of the reads, making appropriate filtering and truncation necessary. Second, sequencing errors and chimeras produced during PCR can interfere with the inference of true sequence variants. DDA2 can address this by building and applying an error model. Third, we must also consider the risk of contamination introduced by reagents, handling, or the environment. Negative controls can help us monitor and assess contamination, but they do not automatically remove contaminants from the data. The removal of low-quality read tails is called truncation. We first examine how quality is distributed across different read positions and then decide where to truncate the reads. Retaining too much low-quality sequence may increase errors, but truncating the reads too aggressively can also remove information needed for taxonomic classification and sequence variant inference. Because this course uses single-end data, there is no need to consider the overlap required to merge paired-end reads. The key is to use the actual quality plots to strike a reasonable balance between reducing errors and retaining enough sequence information, rather than applying a single fixed answer. Next, we will use the single-end denoise single workflow to see how DDA2 processes noisy reads. It begins with quality filtering and truncation, then learns the pattern of sequencing errors from the data. Using this error model, DDA2 identifies and removes chimeras. Finally, it counts the reads assigned to each ASV in every sample, producing the feature table needed for downstream analysis. After running DDA2, we obtain three important outputs. The first is the feature table, which records the read count for each ASV in each sample. The second is the representative sequences file, which stores the actual nucleotide sequence corresponding to every ASV. The third is the denoising statistics, which show how many reads remain after each stage, from the raw input through quality filtering and denoising to chimera removal. These statistics help us determine whether an unexpectedly large number of sequences was lost at a particular step, rather than looking only at the final number of ASVs. Once we have the feature table, community analysis can be divided into two distinct questions. The first is how diverse is each individual sample? This is alpha diversity. The second is how dissimilar are different samples? This is beta diversity. Alpha diversity focuses on richness and evenness within a sample, while beta diversity uses distances between samples to describe differences in community composition. Although both begin with the same feature table, they answer different research questions and cannot replace one another. When evaluating alpha diversity, no single metric is appropriate for every research question. Observed features counts how many ASVs are observed in a sample and primarily reflects richness. Shannon index considers both the number of observed groups and how evenly they are distributed. Simpson type indices are more sensitive to common and dominant groups. Keep in mind that different software packages may use the Simpson index or one of its transformed forms, so the direction of the values may not always be the same. In addition to selecting a metric that fits the research question, we must clearly state the exact definition used. When choosing a beta diversity metric, we can begin with two questions. Should feature abundance, that is, the relative counts of the ASVs, be considered? And should phylogeny, meaning evolutionary relationships, be considered? Jacquard focuses on whether groups are present or absent and uses neither abundance nor phylogenetic information. Bray-Curtis incorporates abundance but does not use a phylogenetic tree. Unweighted Unifrac uses phylogenetic information together with presence and absence, while weighted Unifrac also incorporates abundance. Because these metrics emphasize different aspects of the data, they may reveal different patterns of community variation. In some diversity workflows, including certain analyses in KIME2, samples are subsampled to the same sampling depth through rarefaction. This is one standardization strategy, but it is not required for every analysis and is not the only available method. Setting a higher depth allows each retained sample to contribute more reads, but samples below the threshold will be excluded. Setting a lower depth retains more samples but discards more of the reads that were originally available. We can consult the alpha rarefaction curve and then consider the study design, number of samples, and read distribution together when selecting a reasonable depth. After calculating beta diversity, we obtain a distance matrix describing the relationships between samples. Principal coordinates analysis, or PCOA, projects these distances into a lower dimensional space, helping us observe the structure of the communities. However, points that appear separated on a plot do not necessarily indicate a statistically significant difference between groups. We also need a distance-based permutation test, such as PermaNova. At the same time, we should examine dispersion or within-group spread and consider confounding factors such as batch, individual, and time point so that other effects are not mistaken for a genuine group effect. Next, we come to taxonomy or taxonomic annotation. A reference database stores known sequences and their taxonomic labels, while a trained classifier is a classification model built for a particular target region and reference database. When we feed representative sequences into the classifier, the output is a taxonomic inference, not a directly measured species identity. The taxonomic level that can be supported depends on the target region, sequence information, reference database, and classifier. Only after combining the taxonomy results with the feature table can we create a taxa bar plot. What the plot usually shows is relative abundance, not the absolute number of cells. Finally, evaluate the analysis with 5 questions. Are the metadata and FASTQ files correctly matched? Are quality filtering and truncation settings appropriate? Are DDA2 outputs and retention rates reasonable? Do the alpha, beta, and taxonomy methods answer the research question? Could sequencing depth, contamination, dispersion, or confounding factors affect interpretation? These checks make microbial community conclusions more traceable and reliable.